short list. It's a short list.
Okay, but we need to know. Okay.
Yeah, yeah.
Please,
I make a list for you.
please.
Right. Hello, Abby. Right. I think that is a full house for today. So welcome to Neuroccino, 9:30 on a Monday morning, where we discuss exciting science that you find either in scientific papers, books, book chapters, or anywhere else, and discuss it to the best of our knowledge. Today we have Marcella present, currently from Marseille. And she's going to talk about the semantic encoding at the single cell level. Marcella, if you can share the link with everyone.
Yes. Check where I have all the link. Yeah, this one.
You should be able to share your screen as well.
Yeah. So here's the link.
Lovely. Thank you. Take it away.
Okay. Let me share. Okay, here. Where is my PDF? Is here. Okay. Can you see my screen? I'm trying to make it full screen, but I don't think I can do it. Okay, but can you see it well? It's fine?
Yeah, it's fine.
Okay, good. So yes, hello everyone. Yeah, today I'm gonna share with you this paper I found that was published in July this year, that is entitled, "This is Semantic Encoding During Language Comprehension at Single Cell Resolution." So this paper tried to understand how the meaning of the words are represented at the neural level, right, at the cellular level, and over the time scale of action potentials. And so the aim, well, that was the aim of this paper. And what they did, they recorded from single cells in a particular region of the brain that was the left prefrontal cortex. I will immediately go through all the methods, but it was recorded in the left hemispheres of the brain. And while participants were listening to semantically diverse sentences and naturalistic stories. We are going through the methods here. I'm going down. So here they were patients or participants that had a surgery planned. So they had already electrodes in the brain. So they recorded 133 well-isolated single units in 10 participants across 18 sessions. These participants were 8 male and 2 female, and using this method of this kind of electrodes, tungsten microelectrode arrays. And then in additional 3 participants, they recorded 154 units across 3 sessions. And this was in 2 male and 1 female individuals. And for this one, they use another kind of electrodes, silicon neuropixels arrays. I'm not very expert on this, but I think it's a different kind of recording that you can do with these 2 different types of arrays. And all participants were right-hand dominant native English speakers, and they had normal language that was measured by test before the surgery. What else? Yes, so as I told you before, they were listening to naturalistic sentences, and in total, they had an average of 459 unique words and across 131 sentences. So it was a long experiment across a lot of sessions. And so I think that's all for the methods for now. And what we can see now in the figures is here. So I don't know if the size is okay. Otherwise, you can let me know. But we can see here in A, in the orange, the dark orange, is the places where they recorded the different neural activity. And it was for the 13 participants, and then the lighter orange. So here we can see 2 points. I see 2, but there should be 3. It's where they recorded the other 3 participants. And so here we can see only the activity of the different neurons that they recorded. And so for the material, as I said, they used several materials because they also have a lot of different controls or different experiments. So I will here give you a very overview of all the main experiments they perform. So here in B, we can see the representation they do of the alignment between the onset of the words and the different action potential of the single cells. And what they did for exploring how the different meaning of the words were represented at the neural level, they didn't use, of course, the specific words, but they used this embedding space that we can see in C, where they identify the words or the distribution of the words based on their semantic distance. And they, using a clustering approach, they identify different domains or semantic fields where each of the words were very close to each other at the semantic level, of course. So at the end, in D, we only can see the representations of how well this clustering approach and the clustering algorithm they selected was the optimal one. So they found 9 clusters in total. And yes, that's like how they use all the material and how they explore the data. So now the results, what they found. So in E, we can observe that they identify neurons that responded to specific semantic domains. So here we can see this representation of the results. So here is the result for one of the cells, number 24, which responded specifically to the words that were related to or were part of the food and it didn't respond to any other semantic domain. So here we can see in red the response of this neuron when listening any food-related word versus in green or gray, black or gray, when the participants listen to words related to any other domain. So here in the right, we can also see for another cell that responded to objects compared to other domains. So in total, they observed that the most specific, the largest number of specific neurons responding to specific domains were responding to actions. But here what we can observe is that they identify several neurons, but it was not a lot of neurons. I need to check where I have this information. Yes, it was the 14% of the neurons that responded selectively to a specific domain. But we can observe here that most of them responded to action, then to food states, but also they observe a number of neurons that responded to 2 domains that we can see here in F with the I don't know, the orange squares. No. So most of these, to identify if this the specificity of response of these neurons, they use this selectivity index that one will indicate that these cells respond very specifically to one semantic domain, and an index of 0 will indicate that this response is not specific at all. So this is an overview of the first part of the result. So they identify these neurons that responded specifically to semantic domains. So then they ask, okay, what, oops, I cannot move here. Yes, so they wanted to explore if using the combination of activity of all these neurons that were specific to one semantic domain, if they are able to predict what is the domain of a word that is presented that was not seen before. So they train the neurons, with all the activity of these specific neurons, and then they try to identify which word is the one that is presented to the participants based on the neural activity of these participants. So here in A, we can see that they were able to predict each of the words, or most of the words, provide in a new sentence, not the word, but the semantic field of the word. And here in the right of the A, we can observe that in blue, the correct prediction of the semantic field that compared to chance that is in orange. And this was done using different embedding space. So using word2vec and GLOVE that are different type of embedding for the semantic semantic relationship between words. And it was correct predicted using these two types of embeddings. So this is for the predictions. And then what they wanted to explore was, okay, what if this was specific to the meaning of the words or not? And what they did, they also explore these neuron activities using homophones. So that are words that sound the same, but has a different meaning. For instance, son, so the son, or son, like the child. And so here we can see that the activity of neurons that responded to these words was different in homophone words when compared to neurons. Here we can see in blue that are part of a same semantic field. So it means that the neurons respond in a similar way to words that are part of the same semantic field, but they don't respond similar if the words are not part of the same semantic field, but even if they sound the same. Here in the right of the C, we can also see is a different way to represent the result, but is exactly the same, that the homophone, the difference in the activity between words that are homophones are more different compared to non-homophone words. The third experiment they ran to them was to explore whether these activations that were specific to semantic fields was dependent or not of the context. So what they did, so they compare the activation of these specific neurons within these sentences that they presented to the participants, but they compare with sentences where the words were in a random order. So we can see here, I don't know if you see in D, below, it's the bike called child. It's really a list of words that is not a real sentence. So what they observed is that we can see in the upper part of the D that the single cells responded specifically to these words that belong to the semantic field of family, but then in this case was for the word child. And then below we can see when the word is provided in a list of words, it didn't respond to this word. So what these results suggest is that these specific neurons respond to these specific domains, but only when they are provided in a context. And here we can see in E, the index of selectivity, so the specificity of response of these neurons to a specific semantic field when they are presented in sentences. So in this pink color, I think, or orange, and that is closer to 1 because it's not a super high index. All the time was 0.4 max. And we can observe in brownish or gray that it's this index decrease when they are presented in this word list. So this is for the context, and I think the last this in this is also for this figure, the 3, it's also reaffirming the results that I already provide. So here in A, for each of the participants, from 1 to 10, they show the difference in activity when they are presented with these homophone words compared to these that are two words that belong to a same semantic category, where the difference in activity is lower. And in B, what they demonstrate is that the prediction of the word or the semantic field of the word that the participants are listening, it's better predicted when it is a low surprisal. So when you know that, like when the word is provided in a semantic context that is related to the word. So I don't know if it's clear, but it's if you expect that this word will be has been provided before with these other words, it's better predicted. I hope it's clear. Otherwise, you can let me know later. And this is the last figure, the last result that I will share with you. So this, okay, based on the previous results, they wanted to know if the neurons may represent the semantic relationship between words at the neural level. So if they can observe based on the activity of the neuron, if they can identify a representation of the semantic relationship of these words. So what they did, they regress the responses of the neurons onto the embedding vectors of all the words that they studied that they presented to all the participants. And it results in a model with the weight of the neurons. So how each of the neurons contribute to the representation of each word. Okay, so this is a little more complicated for me to explain, but what I want to show you is that after they created this model, they observed that, well, they use principal component analysis, where they identify 5 principal components that explain the variability of all neurons in a 45%. And specifically for the selective neurons, so the ones that are specific to semantic fields, they explain the variance of this activity in 81%. So what they show here is that there was a direct relationship between the activity of the neurons and the semantic distance of the word in this embedding space, which can suggest that the relationship between the words, so the semantic relationship between the words, are represented at the neural level when we compare the activity of the neurons when these words are presented. I think that's it that I want to share with you. So then they have the discussion that, of course, it was specific to the sentences. So also they, of course, provide the importance of the context to the observation of these specific responses of these neurons. But of course, it's still unclear if it's generalized to other modalities, or even to non-linguistic stimuli, videos, picture, etc. And also that it was only recorded into the prefrontal cortex. So it's very specific to the prefrontal. And also that was important, I think it's important that it's not mentioned here in the discussion, is that this specificity, so the amount of neurons that they capture, only the 14% were specific to semantic domains. So it's not a lot, but still it's a result. And yeah, I'm happy to take your questions if you have questions or comments, or if I can reply, of course.
Thank you, Marcela. I missed the last numbers. How many % are specific to semantic?
14.
14, 1, 4. Okay, yes,
Yes.
really, really not that much.
Yes, yeah.
Eduardo has his hand up first.
Yeah, first.
Yes, thank you, Marcela, for presenting. I have a bunch of questions. So the first one, I couldn't see. Easy question. What kind of patients they study, like epileptic patients? Because I couldn't find any information in the paper just by like screening.
Yes, it's here. It didn't say it's epileptic. They say that they were patients that were ready to have neurophysiological, yes, but it didn't say which kind of specific patients they are. Yeah. Also, they perform analysis using the different clinical diagnoses, but I don't see that information.
No, okay. The second is whether they use some theory of semantics as a reference to build some hypothesis, because also I couldn't find any reference just by going quickly through the text. So maybe since you read
they use
it.
What? Sorry.
So is there any theory of semantics they use to, build hypotheses, or because I quickly went through the paper, but I couldn't find any reference to any specific theory. So I was wondering if maybe I missed that.
No, I didn't. Of course, I went through all the methods and also to the supplementary material or extended data. I didn't find it either. There is most of the they base their analysis on this embedding space they built at the beginning using this I think it's word2vec, but yeah, it's here in C. So they created this space, and using this, they only take the semantic distance between the words using this clustering approach. But then to interpret data, no, there is no theory of theory they mentioned.
Okay, yeah, thank you. And yeah, one last question is related to these embedding spaces they use. Is there any reason why, for example, in the PCA analysis they use what was that, like 5 components to analyze the variance, or is that data driven, theoretical, I guess not driven, but yeah, just to know if there is more information on that.
I don't remember if they mentioned, maybe they do in the method, but it was data driven. I guess they selected based on the results, this elbow thing, but I don't remember exactly. But here, finally, efficiency was used. No, but you can go through the method. Is here in the on page. I don't see the number of the page.
Yeah, no, it was just out of curiosity.
But yeah, it was data driven based on their analysis. I guess they took the ones with the, I don't remember, Hagen value higher than 1, something like that. I don't remember the name.
Well, I would like to see a script plot somewhere, but yeah.
Yeah, exactly.
It's not there.
No, they are no. It's not in the extended data either, because I went through it. But you can also double check. Yes, there is something in the extended data figure 1, but it's not for that analysis. Sorry.
Okay, yeah.
But
okay,
you can
no
check.
worries.
Probably you can find something.
No, yeah, thank you for the presentation. It was really dense paper.
It was.
Miguel was next.
Yes, thank you, Marcela. It's very interesting paper. I had a couple questions. So the first one is the posterior portion of the middle frontal gyrus. Like, did they have any expectations about seeing something like this in this region? Like, I don't know what this region's role in language processing. So it's
I don't think, at least in the introduction or in the discussion, I didn't find a reason why they selected the prefrontal. I guess, but it's really my guess. it's probably they had this opportunity, they had the patients with this part, like of the brain that was interesting for exploring language. Maybe that's why, but they don't specify why frontal compared to others regions that are also related to language. They mentioned that as a limitation, that of course they only capture the activity in the prefrontal, so of course it remains to be explored what happened in other regions. But yeah, I don't remember seeing any hypotheses or yeah.
But the task the patients are performing is just they're just listening to sentences?
Yes. Yes.
Okay.
Yes, just listening.
It's also interesting to me that you said when they were presented with like switched words, they don't see any differences between like semantic categories, but this is only when these words are embedded in a sentences. So that part also, like there are different controls that they could have done. For example, they could give the word list just as a word list, not embedded in like a sentence structure. Because what I'm thinking is some of these neurons may also be responding to the prosody of the sentences, right? So to intonation, or as another control, they could flatten the intonation. So are there any, like I couldn't come across, but are there any controls that other than like switching the word order in a sentences?
No, and no, that I remember. No, I have all here. So the sentences, they also, no, for the predictions, they also made the predictions in narratives. But yes, same also they had the prosody. And then across individuals, across techniques and patients. But no, the control, no, they also explore with non-words, but it was different for a different questions, not for this one. But no, for control, it was only the the word list. I'm just corroborating what to say something that is not correct, but yeah, it was linguistic material where the sentences, the stories, word list, and non-words. And the word list were these only
They are
without
still in a
context.
sentence structure, yeah, that's interesting to me. Like I was wondering what their interpretation is. Like the semantic category doesn't apply to word lists, but only when it's within a sentence.
Yeah, I guess with the word list, I guess it's not explicit, but I guess they removed the prosody. I guess it's only like word list, like really. But I don't know, maybe I miss it, and in the method they provide some information about that. But I in what I remember, I didn't see anything specifically related to this. So
okay, yeah, it's okay. I'll check that. Okay, thank you.
Yeah.
Great questions and comments so far. Just a very quick one. So from stroke and dementia, we know that the temporal lobe is quite important. I was wondering if there's any discussion on the role of the other lobes in semantics, because there's theories about the parietal lobe and the temporal lobe and a lot less about the frontal lobe.
Yes, I was also surprised because when I found the paper, it was really because of the title. So I was really, wow, this is very, but yeah, it was limited to the prefrontal. They say it as a limitation, but they don't mention or they don't discuss other regions. They are very, the only limitations is we should explore the other regions that are related to language, but for the discussion of the results, they mostly focus on what they found and mention a little bit. It say that this is very restricted to prefrontal regions and that they should explore more, but no, nothing, not temporal lobe, no diagnosis, not patients, nothing of this. It's in the discussion.
There's also a question on YouTube about the definition of semantics, questioning if semantics is rightly defined in this paper.
No, I'm really don't want to make mistake, but no, I don't think they define. I guess, yeah, they say our semantic knowledge is highly structured, but yeah, there's no definition. But I guess because of the method they are using, they really are focused on the semantic distance based on this all embedding space that they use that are, I guess, trained in stories, internet, or, but yeah, there is no definition that I remember that they specifically made for the semantic in the paper.
Thank you. And Michelle has been patiently waiting.
Yes, thank you. I'm sorry if I'm correct. I think that's...
I was just wanted to mention that there was like a region in the frontal lobe reported by Elizabeth Warrington that was supposed to be related to semantics. And thank you for your presentation, but I can't help but wonder whether the interpretation of the paper is like those semantic concepts are localized in those single neurons, or do they really, how do they interpret that? They, you know.
Yeah, yes. Okay. Of course, that at the neural level there is a role that in a context, because of course it's only in a context, that also at the neural level there is this representation of the semantic relationship, but that's it. I don't think, of course, not exclusive. Only they suggest that this is a cellular mechanism that could help to to map the semantic information at the brain level. But yeah, it's real, something that they mention as a very real real time during speech, that this is at the cellular level what's happening to interpret the meaning of a word within a context. But then there is no, at least what I remember, still sorry if I'm mistaken, but there is no other idea about how this is integrated to the rest of the process of language that we know. And but I don't think they say that this is the way, or like this is the region, because of course they only explore this and then explore other regions of the brain. But yeah, there is no discussion about how this will relate to what we know about the comprehension of language within the brain as a network.
That information got to come from somewhere and go somewhere else in order to shine light in these concepts that we have in the brain. But that's my take on this. Thank you.
Thanks, Meet. Final question from Eduardo.
Yeah, one last, sorry, I'm running late, but building on what you just said, I was wondering, so that they investigated like homophones and, well, semantic distance, of course, with these embedding spaces. But looking at the example, I was wondering whether they also consider grammar and syntax, because this is also one piece of information that might be already there in the brain, as Michelle was saying. And you know, the example there is in figure 2, when the sun smiled or when the sun set. Sure, they are homophones, but they are embedded in different grammatical structures. So I was wondering whether they mentioned something about that. No?
No, not that
Okay.
not that
Okay.
I remember, or nothing very explicit and clear in the text.
Yeah,
Maybe in
it
the
just came to my
material,
mind.
but yeah.
Yeah, okay. Nice.
And that's a wrap. We're out of time. Thank you so much for presenting the paper. Thanks for all your questions, and see you again next Monday. Have an exciting week in science. Bye.
Thank you. Did you stop recording?